Hello everyone, I'm your teacher Amar. I hope you are fine. So before starting the video, please subscribe to channel and click the bell icon so you will get notified every time I upload a new video. And like this video. In this video we talk about recombinant DNA. You know human have 46 chromosomes, 23 pair, 2 meters of DNA, 3 billion DNA subunits like the bases ATGC and 30,000 genes that code for proteins. Okay. DNA technology involves genetic engineering that can be used to cure different sorts of diseases, genetic disorder, and to improve food crops. These are the Tasmanian tiger, herbaceous. This is the Dolly sheep. Okay, a restriction enzyme is a bacterial enzyme that cuts long strands of nucleotide into smaller segments that can help to manipulation of DNA. We have to recognize specific sequence of nucleotide and then cut the DNA specific side within the sequence. In one chain, the sequence runs left to right, and on the complementary chain, sequence runs right to left. So 3 to 5 and 5 to 3. Okay. These are the restriction endonucleases that cut DNA at specific sequence and they recognize palindrome. EcoR1 is the enzymes and these are the restriction enzymes. EcoR1 unmethylated DNA and they cut cleavage from 5 to 3 and 3 to 5 and create a sticky ends. Okay. Single chain tails of DNA called sticky end are created on each DNA segment by the action of the restriction enzyme. Thus, the sticky end readily bind to the complementary chains of DNA. When the piece of DNA that have been cut within the restriction enzyme, they can be bind together to form a new sequence of nucleotide. And restriction enzyme can be used to isolate cut and specific gene. By cloning vector, we have to, these are the basic carrier which is used to transfer of gene of interest. DNA carrier used to clone a gene and transfer it from one organism to another. Once gene has been isolated, that small piece of DNA can be placed into cloning vector can be introduced into organism. The ligation is used by ligase enzyme. Many bacteria contain cloning vector called plasmid. The DNA fragment to be cloned enzymatically inserted into DNA fragment. These are the plasmid from bacteria, and these are the our cloning vectors with the gene of interest, and they can be transformed E. coli cell and select ampicillin resistant cloning. After transformation, we can select which colony has our gene of interest by the selectable marker. In this case, is the ampicillin resisting colony. Plasmid, a ring of DNA found in a bacteria in addition to its main chromosome, it can be used to transport genes from one organism to another. Okay. This is the electron micrograph of plasmid. This is the diagram of plasmid. Okay. Moving on to the next slide. Process of using a cloning vector. Plasmid is first removed from bacteria cell using restriction enzyme. Plasmid is cut and donor gene is sliced in. Donor gene, a specific gene that is isolated from another organism and introduced into the cloning vector. This is diagrammatic representation. This is our plasmid with EcoR1 enzyme used to cut the specific area of our vector. This is the sticky end of DNA to be inserted. EcoR1 enzyme is used. After that, we have to ligase both into a recombinant DNA. This is the cloning vector with our gene of interest, and then it is inserted into the foreign or host organism. Plasmid is returned in the bacterial cell where replicate as bacterial cell divides this clone the donor gene. The bacteria containing clones of the donor gene can be then be used to infect other organisms and transfer the gene to them. Okay, plasmid vector DNA fragment to be cloned enzymatically inserted into DNA plasmid vector. This is the recombinant plasmid with our gene of interest in the black. We have to mix E. coli cell with the plasmid in presence of calcium carbonate, culture on agar media, and having ampicillin. This is the transform equalized cell survive, but the without transform or without gene of interest cannot transform. We can make copies of the our survival bacteria. Transplanting gene in some cases, plasmid can be used to clone specific genes so that bacteria will produce a specific protein. Insulin is the core example where it controls sugar metabolism. This is the diagrammatic explanation of the insulin gene resistance transformation. Restriction enzyme, same plasmid gene of interest, it is into the transfection is done, and the protein insulin gene is encoded into bacteria that produce insulin hormone protein. Transplanting gene. People who are diabetic do not make hormone insulin. Insulin gene allow sugar to travel from blood into the cell where they are transformed into the energy. A large volume of insulin can be produced by cutting out the human gene and cloning into plasmid process. Isolating gene, same DNA is removed from human cell to isolate gene that produce insulin. Okay, restriction enzyme used to cut the human DNA into separate genes that include gene for insulin, and then producing recombinant DNA, combination of DNA from two or more sources, inserting donor gene, human gene, insulin into bacterial plasma, resulting in recombinant DNA molecule, and then cloning DNA, then plasmid containing recombinant DNA is inserted into host bacteria. The transgenic bacteria can be placed in neutral medium where it can grow and reproduce. Within each bacterium, the plasmid is copied many times and donor genes for insulin is cloned. A thousand of bacteria produce very quickly through mitosis. The transgenic bacteria can be used to produce large amount of insulin. And same, this is the diagrammatic explanation of production of insulin. Expression of cloned gene. Once a donor gene is transferred to the host cell, it is transcribed and translated as though it were in its own cell. Not all of the genes in cell genome are expressed. Genes are often turned off until the protein they code for are needed. DNA fingerprinting. The bending pattern of DNA fragment from two different individuals may be compared to see if they are related. For example, paternity testing. DNA fingerprint of member of two different species can be compared to determine how closely the species are related to find the evolution history, and using DNA fingerprinting to compare sample of blood, hair or tissue found at crime scene with the suspect blood sample, which may help in solving crime. Okay. Making a DNA fingerprint, RFLP restriction fragment length polymorphism analysis method for preparing a DNA fingerprint involve extraction DNA from blood, hair tissue, cutting it into fragment using restriction enzyme. Fragment of DNA are then separated using a technical gel electrophoresis. This is the diagrammatic representation of gel electrophoresis where the DNA fingerprinting is carried out. Making the gel electrophoresis separate nucleic acid or protein amino acid according to their size and charge. Sample of DNA being compared or placed into wells made on the gel and electric current is run through the gel. Okay, this is the diagrammatic explanation of the DNA fingerprinting and how the gel is run. Okay. And DNA fingerprint negative charge migrate towards the positive end of the gel. The smaller DNA fragment migrate faster and further down the gel than the longer fragment. The DNA fingerprint that have been separated and gel are stained in order to be seen. We have these are the stained particle of DNA. We can see them DNA A, DNA B, label them. And accuracy of DNA fingerprint. The complete included sequence of each individual is unique for each person except identical twin. Therefore, everyone DNA fingerprint will be different. DNA fingerprint are 99.99% accurate. Polymerase chain reaction PCR can be used to make copies of the selected segment of DNA for. So the PCR it is used to make copies of selected segment of DNA for a DNA fingerprint that require primer, that is a artificially made single-stranded sequence of DNA. When these ingredient DNA primer are combined and incubated, the selected region of DNA can be quickly doubled. This is the PCR diagrammatic explanation. Okay, every 5 minutes the sample of DNA double again, resulting in many copies of sample in short span of time. The new copies of DNA sample can then be used to make a DNA fingerprint used in paternity test, diagnosis of genetic disorder, study of ancient fragment of DNA. Human Genome Project goals to determine the nuclear sequence of human genome and to map the location of every gene on the each chromosome. To compare the human genome with the genome of other organisms in an effort to provide insight into the fundamental question about how genomes are organized, how cellular differentiation and growth are under genetic control, and how gene expression is controlled and how evolution occurs. It is hoped that knowledge gained from human genome project will improve diagnosis, treatment, and even provide cure for genetic disease. These diseases can defective protein can be cured, identified, and can be coded, which make possible to design therapy aimed at correcting gene defect responsible for the disorder. Gene therapy and cloning. Treating a genetic disorder by introducing a normal gene into a cell or by correcting a gene defect is called in the cell genome is well suited for treating genetic disorders that result from deficiency of single enzyme or protein. An example cystic fibrosis, lung cancer, AIDS, ovarian cancer, brain cells. Many medicines are proteins that can be mass produced in less expensive way through DNA technology. Examples are insulin control, sugar metabolism used to treat diabetes, clonal stimulating factor used to treat immune deficiency by stimulating the production of white blood cell, erythropoietin used to treat anemia by stimulating production of red blood cell. Human growth hormone used as treatment for dwarfism causes bones to elongate. Interferon used to treat viral infection and cancer by preventing the replication of viruses. And interleukin used to treat HIV and cancer by activating the and stimulating different kinds of white blood cells. Genetically engineered vaccines, many diseases are combated by prevention using vaccine made by genetic engineering, increasing agriculture yield. DNA technology has been used to develop new strains of plant which in turn can be used to improve food and crop yield. For example, tomato and worms by transferring genes for enzymes that are harmful for hormones into tomato plant. Scientists can make tomato plant toxic to hormone and effectively protect the plant from these pests. Okay, they can decrease the yield. So in order to increase the yield, we can engineer a new tomato variety. GMO, wheat, cotton, and soybeans have been created to resistance to weed controlling chemical. Herbicide, weed controlling chemical. Such herbicide resistance crops can protected from weeds more easily and less expensively than crops that are susceptible to these. The example given above are also classified as therapeutic cloning. Therapeutic cloning used genetic engineering to help treat and cure diseases. Okay, safety and environmental issues that are a core concern. Many people are concerned about safety of genetic engineering food. The concern is that food production by GMO can contain toxic protein or substance that can cause allergy in people who consume them. Food produced by transgenic crop can be sold without special permit or label if the product is identical to product produced by non-transgenic crops. Example corn, tomato. Concerns: GMO crops should be separated into wild and wipe out native plant species. Transgenic crops could transmit their new genes to other species in neighboring area. For example, superweed produced by rice and long grasses exchanging pollen with native species. Reproductive cloning involves making a whole new organism from a still living organism. There is no sexual reproduction or fertilization involved. Sheep, donkey, and number of plants have been cloned. Human cloning is still far off in the future. And these are the steps in the reproductive cloning. That's all from today. I hope you like the video. Please, if you haven't subscribed, subscribe it. Click the bell icon for the future notification and like the video. Thanks. I will meet you in the next topic.